we have on the screen the objectives, the study objectives from Chapter 7. And today I'm going to probably in 2 lecture segments because this software records 15 minutes at a time. But in 2 lecture segments, I'm going to go over very likely the first 5 of these study objectives. So this is what we're focusing on. Of course, there is some vocabulary associated with this chapter, and I will be highlighting those things as we go through. But also I'm going to ask you to memorize the summary equation of cellular respiration. You're going to need to be able to name the 4 stages of aerobic respiration. And then we're going to prepare, and you're going to put into your notes a chart that shows all of the things that are on this study objective here, the output, in really reactance and output of each stage. And then we'll talk about the cycle that's called the Krebs cycle and what the defining criterion of that term is. And we'll talk about pyruvate, the product of pyruvate, how it's prepared for entry into the Krebs cycle. So these 5 are the focus of today's 2 lecture segments, and you're going to take very careful handwritten notes. All right, so the first slide says life is driven by energy. As we talked about in Chapter 6, energy is usually defined as the capacity to do work, although I typically don't find that particular definition that helpful, but it is the thing that flows through a system. In this case, we're talking about a cell, but also through a whole organism, even a whole ecosystem. And as energy flows through a system, it is transferred and transformed by what we call coupling. So reactions that release energy are coupled to reactions that absorb or require energy. And so in this slide, we define 2 types of organisms. Autotrophs, those are photosynthetic organisms. They're able to make organic compounds by a process called photosynthesis. That happens to be the topic of our next chapter, Chapter 8. And heterotrophs have to get organic molecules from autotrophs, and we mean they eat autotrophs to take those organic molecules and use them for themselves. But all organisms, whether they're autotrophs or heterotrophs, use a process called cellular respiration. So it's a metabolic pathway, which you'll remember involves a series of enzymes acting in a sequence to ultimately transfer the energy from the bonds of organic compounds. Remember, covalent bonds are energy, they're potential energy, into the covalent bonds of a different molecule called ATP. So also a type of potential energy. And so that's what cellular respiration is at the fundamental cellular level. So potential energy is stored in organic molecules in the covalent bonds, particularly in the carbon to hydrogen covalent bonds. And we know that carbohydrates, proteins, and fats have these covalent bonds in them, and they are what holds that potential energy. Reactions that break down these carbon to hydrogen covalent bonds are called oxidation reactions. So often cellular respiration is referred to as the oxidation of organic compounds, or even more specifically, the oxidation of glucose, because glucose is the kind of the quintessential organic compound that's always used as the main example, although we'll see how other compounds can also be oxidized. But the oxidation of glucose, the oxidation then of amino acids, and the oxidation of fatty acids, this breakdown of these covalent bonds releases that bond energy. And so those are exergonic reactions, and they release that energy from the covalent bonds. When that energy is released, that release of energy, that exergonic reaction is coupled to the endergonic reaction, which is ADP, adenosine diphosphate, plus inorganic phosphate combined together in a synthesis reaction to make ATP. So that synthesis of ATP is endergonic. The oxidation of organic compounds is exergonic, and so the two can be coupled. And ultimately, that is what cellular respiration is, the coupling of the exergonic breakdown of organic compounds to the endergonic synthesis of ATP. It may not be what comes to mind when you think of that word, respiration, but fundamentally, that is the important part of it. So what is oxidation? Oxidation, from a purely chemistry standpoint, is defined as the loss of electrons. There's an opposite process called reduction, which is defined as a gain of electrons. And the reason reduction is a gain of electrons is, if you remember that electrons have a negative charge, then when a molecule gains electrons, its charge is reduced, becomes more negative. So that's where that term reduction comes from. So oxidation is the loss of electrons, reduction is a gain of electrons. And so the two go hand in hand. If one compound loses electrons, there has to be a compound somewhere else that gains them. And so as the electrons are passed from one compound to another, so is the energy. So it is a loss of electrons, but also a loss of energy, and a gain of electrons, but also a gain of energy. Dehydrogenation is a loss of a hydrogen atom, which you may remember is composed of one electron and one proton. So dehydrogenation is sort of a special kind of oxidation. It does involve a loss of an electron, but also a proton, so it gets a special name. But in any case, in cellular respiration, as we're talking about energy being transferred from one molecule to another, what we're really also talking about at the same time is electrons being transferred from one molecule to another. Those go hand in hand. And so sometimes you'll see the term redox. A redox reaction would be a coupling where one compound is oxidized, loses electrons, and the other compound is reduced or gains those electrons. So it's just a transfer of electrons from one compound to another. So during redox reactions, the electrons carry the energy from one molecule to another. So as electrons are being lost and gained, so is energy being lost and gained. You can consider those things connected. There is a middleman carrier in this process. It's called NAD. And the reason it's called NAD is because the real name is listed here, nicotinamide adenosine dinucleotide. And NAD is just easier. And so we call it NAD. It's an electron carrier, but honestly, it's really a hydrogen carrier. And remember that within the hydrogen, there's an electron. So when NAD, or it's really NAD plus because it has an extra positive charge, when NAD plus picks up 2 electrons and 1 proton, the proton is just sort of a tag along. It's not the important piece of this, but the electrons, it becomes NADH. And so we say that reaction is a reduction reaction because the NAD is gaining electrons, and so it's called reduction. However, this reaction is reversible. The NADH can pass those 2 electrons and protons on to some other molecule to become NAD plus again. So really the NAD is picking up electrons from one compound and then carrying them and giving them away to another compound. So that's why we call it an electron carrier. It's just a pickup and a drop off in the middle of this series of reactions that we're about to talk about. And here's the structure of NAD and the structure of NADH, which you do not need to memorize. But you may have noticed that in the name NAD, the D stands for dinucleotide, And I think you guys can recognize the two nucleotides connected together that make up the main part of this molecule. And then there's some other stuff up here. But anyway, so when the NAD plus doesn't have its two electrons, it would have the structure shown on the left. When it does have the two electrons and a proton, it would have the structure on the right. But I mean, it's very minor difference, visually, and you don't have to memorize the structure of it. So I just wanted to kind of show you that dinucleotide structure as a throwback of something you've learned before. Okay, so cellular respiration. Overall, what cellular respiration is like I said, it's the transfer of energy from the oxidation of glucose, from the covalent bonds in a glucose molecule or other organic compounds, into the transfer of that energy into the covalent bonds of ATP. And so as the electrons are carrying the energy, it's not just the transfer of energy, it's the transfer of electrons. So you have dozens of these redox reactions, dozens of these electron transfers taking place. And there are going to be a lot of electron acceptors and electron carriers involved. The main electron carrier is NAD+. But in the end, I'm giving you an overview here. In the end, all the high energy electrons, the electrons from the covalent bonds of glucose or fatty acids or amino acids, but all those electrons that have all that energy are going to lose most of their energy. Their energy will be passed on somewhere else. And the electrons, at that point, they'll be low energy electrons. Once they lose all their energy, will be transferred to what we call the final electron acceptor. There are three types of respiration that are really defined mainly by their final electron acceptor. At this point, that's how we're going to define them. And it is really the first way that we define them. The one we're going to talk about the most is called aerobic respiration. Aerobic respiration, the final electron acceptor, which I haven't really explained what that is yet, but right now we're going to just say the final electron acceptor, whatever that is oxygen. So aerobic means with oxygen. Anaerobic involves a final electron acceptor that is some kind of molecule that's not oxygen. But it has to be inorganic, meaning not a carbon compound. So a non-carbon compound, but also not oxygen. So often anaerobic respiration, the final electron acceptor is sulfur, for example. Fermentation is quite different. It's where the final electron acceptor is an organic molecule, some kind of organic compound. And the one we're going to look at is a molecule called pyruvate. We're only going to touch briefly, though, on the anaerobic and the fermentation pathways. Most of our chapter is going to be focused on aerobic respiration, so respiration with the final electron acceptor that is oxygen. So the last slide for this segment of the lecture is the overall equation of aerobic respiration, which is typically given by the top equation, C6H12O6, which is glucose, plus 6 oxygens, because this is aerobic respiration, yields, that arrow means yields, 6 carbon dioxides and 6 waters. But that arrow, that yields arrow, there's a lot of steps in that. So this is just the very beginning reactants and the very end products. And we're going to delve a little bit into some of the middle steps. But this equation just summarizes the overall. Now, this breakdown of glucose releases, delta G is a negative number, 686 kilocalories per mole of glucose. Now, that number doesn't have to mean anything, but the fact that it's a negative delta G, it tells you that it's releasing energy. But the energy is not released all at once. It's released in little increments. And then the real equation, which we'll start in the next segment with this bottom part of this slide, involves the building of ATP. So that's next.